This video is going to be a brief background for our Yeast Crispr CURE. The first lab, the cloning. So first, let's give a really brief background about restriction enzymes. So these are enzymes that recognize specific sequences of DNA and then cut both strands of DNA. The restriction enzymes, they recognize what we call palindromic sequences. That just means the sequence is the same backwards and forwards, essentially, for these complementary strands. So if we have a sequence here, TGA, TCA, if we went to the other strand and went the same kind of 5' to 3' direction, it's also TGA, TCA. So in that sense, this sequence is a palindromic sequence. And these restriction enzymes will recognize some specific palindrome, and there are many different restriction enzymes. And originally isolated from microorganisms. And so here's an example of two different restriction enzymes, a SWA1 or SMI1 restriction enzyme, and it's BCL1 restriction enzyme. And in one case here, we have the restriction enzyme cleaving those strands to make what we would describe as blunt ends. In the other, you can see it's cleaving both strands and yielding what we would call sticky ends. So these ends have overhangs. In this case, we have a 5' overhang here and a 5' working on this strand. So we have 5' overhangs, and we'll see how that can be useful in a second. And this is just, there'll be no overhangs, so that's what we call blunt-ended. And the other tool we're going to use here is another enzyme called T4 DNA ligase. This is originally isolated from a bacteriophage, T4, and it's a very commonly used kind of molecular biology tool. So if restriction enzymes are kind of our typical cloning kind of molecular scissors to cut DNA, DNA ligases are kind of molecular glue to stick it back together. So if you have a DNA strand that has a 3' hydroxyl and another DNA strand that has a 5' phosphate, T4 DNA ligase can ligate those together, stick those two strands back together, and reform or form a phosphodiester bond between those two strands. And one last really brief bit of information. We'll be using E. coli as sort of our cloning workhorse, which is very commonly done in biology. And E. coli can pick up what we call transformed, meaning they can pick up circular plasmid DNA, these plasmids from blood vectors. So we have a circular piece of plasmid DNA that can go into be transformed into the E. coli, and they can actually stably maintain that plasmid and propagate it to their offspring. And the way that we force this event to happen is that there's typically some form of a selectable marker on that plasmid. For example, ampicillin resistance, probably the most ampicillin resistant gene is probably the most commonly employed kind of selectable marker that's used in E. coli. The ampicillin is an antibiotic that is a irreversible inhibitor of an enzyme that bacteria use to build their cell wall. And so ampicillin inhibits this enzyme and prevents bacteria from growing. So if we have just kind of normal, not ampicillin resistant E. coli, and we grow them in the absence of ampicillin, they will grow really well. Here's kind of represented on this cartoon petri dish. If you put them on a plate with ampicillin, they will die because they're not able to make their cell wall. But if they have a gene that confers resistance to that ampicillin, then if you put them in the presence of ampicillin, some of those colonies will now grow. So you can see here, there's no ampicillin added. A lot of bacteria are being transferred, but only some of those would grow in the presence of ampicillin. And the ones that would grow would be the ones that took up the circular plasmid DNA. And that's our selection there. So we know that each one of these colonies on this plate must have taken up that plasmid DNA. So now they have a plasmid of interest. Okay, so for the Yeast Crispr CURE, the plasmid we'll be using is PML104. This was made by the Weyrich lab. And again, it has the Cas9 enzyme, the enzyme that is going to can cut DNA, kind of like restriction enzymes, although instead of just intrinsically recognizing a palindromic sequence, Cas9 enzyme is going to use this other component, this guide RNA, to tell it where to target. And now instead of the restriction enzymes where the palindromic sequences they use are very small, so they would cut many places in the genome. Again, the guide RNA that the Cas9 uses can be very large, 20 nucleotides, so that it can be very specific and potentially cut, say, one place in the genome. So very precise, much more precise molecular scissors than restriction enzymes. We've got the Cas9 enzyme, we've got the guide RNA, and what we're going to do with this cloning is clone in this targeting sequence of the guide RNA for Cas9. And so that's what we're cloning in. We're going to have some of this plasmid that's been cut with this BCL1 and SWA1 enzyme. And we're going to clone in some small oligonucleotides that will correspond to this guide RNA sequence. This PML104 is what we call a shuttle vector. So that just means it's got the ampicillin resistance gene, so we can do the molecular biology stuff in E. coli. Then it also has a yeast selectable marker, yeast origin replication, bacteria origin replication, so they'll copy these plasmids. And the yeast selection we have is the URA3 gene. This is a gene involved in uracil biosynthesis. The strain of yeast we'll be using is missing this gene. So if you grow them in the absence of uracil, they won't be able to grow because they're not getting it from the environment. They're not able to synthesize itself because they're missing this one gene in this pathway. We're going to put that gene back, and that's how we can do selection, kind of like the ampicillin selection that we just talked about. Okay, and here's just a little bit more detail from one of the YREC lab papers where we kind of see that the plasmid they gave us has, again, this BCL1 site and this SWA1 site. We've cut them with both of those enzymes so that one of these pieces is going to have a overhang, and the other piece is going to, the other part of the plasmid, these will both be connected to the circular plasmid. I'll leave this little piece out. And now we've got a sticky end and a blunt end. And so when we designed our oligonucleotides, we actually used the YREC lab software that sticks on, in addition to having our guide RNA sequence of interest, it's also got just the blunt end at one side and a little bit of the kind of scaffolding part of the guide RNA, and then just that sticky end on the other side. And that sticky end will help kind of direct this piece of our double-stranded oligos that we're trying to clone in the right direction, so that it didn't kind of go in there backwards, because on our plasmid we'll have that kind of corresponding sticky end on the other strand that we're going to ligate that into. And so we'll have the complementary base pairing occurring that will facilitate that binding of the sticky end over to this piece, but not to this side of the plasmid. And then we'll ligate those two pieces together with T4 DNA ligase. And then once we do that, the next step is we don't want to actually validate this by sequencing. We'll just do a colony PCR we'll talk about in another video.